So, biotech.
Better future.
Recombinant DNA technology can be used in the production of human insulin.
You may have heard of E. coli before, but what is it and how can you prevent it from spreading?
There are roughly 100 trillion microbes in our gut, and around 100 billion of them are E. coli. This biological rockstar is a strong pillar of biopharmaceuticals. They have given insulin, growth hormone, and interferons. Their contribution in molecular biology and medical bacteriology is huge, like Luria Delbrück experiment, Meselson Stahl experiment. But then what is this?
The nation's top disease detectives are searching tonight for the cause of a mysterious and deadly new outbreak of E. coli.
Seeing record-breaking levels of E. coli.
The CDC is warning Californians about an E. coli outbreak leaked.
After nearly a month, the prime suspect in Alberta's worst ever E. coli outbreak confirmed.
It is an especially virulent strain, and a number of states are already affected. There is concern this outbreak will spread.
So why do biologists and biotech industries use a species that could cause a fatal outbreak? Is this species good for you or harmful for you? You can find this answer using Google within a second, but let's explore the problem from historical angles. This will help us to understand the methods scientists employ to resolve such problems. The story has three parts, and they are the good, the bad, and the ugly reality of E. coli. So sit back and relax. Let's explore the fascinating history and scientific aspects of this extraordinary journey undertaken by a model organism with a controversial past. It was around 1885, a child specialist, Dr. Theodor Escherichia, who had a good amount of knowledge on microbiology and cholera epidemic, isolated a bacterium that was significantly higher in the colon of milk-fed newborns than adults. He named it Bacterium coli commune.
Coli actually refers to colon, where they usually inhabit.
Later, some scientists also detected this species in the gut of other animals. The bacteria became popular in less than a decade because it was an easy organism to grow under different conditions, had a short generation time, and no specific nutritional requirements were needed. These characteristics made the species a popular choice in the field of bacteriology and molecular biology. Later, the bacterial isolate of Theodor Escherichia was stored in 1920 in the Lister Institute in London, and unfortunately, this isolate did not get explored more. Rather, some other strains like K12, B, C, and W have been established as model organisms. We will talk about these strains after a few minutes. Let's go back to the history again. In 1907, a scientist named Rudolf Machini made an interesting discovery about a certain type of Bacterium coli. He found that this particular strain of bacteria initially struggled to process lactose, the sugar found in milk. However, after a few generations, they developed papillae on their colonies and began fermenting lactose. The strain was named Bacterium coli mutabile. This discovery likely paved the path for numerous subsequent studies on E. coli, ultimately contributing to the rise of molecular biology. During 1919, the writers of Manual of Tropical Diseases suggested changing the name from Bacterium coli commune to Escherichia coli, since Theodor Escherichia first isolated this bacteria. So, Castellani and Chalmers proposed the popular name Escherichia coli for the first time. Many scientists and doctors had developed a strong affinity for this organism. Given its ease of growth, it was utilized for educational and scientific purposes, including the study of medical bacteriology, bacterial physiology, metabolism, molecular biology, genetics, and evolution. Everyone was selecting this organism because there was an abundance of information available on this bacterial species compared to others. During the 1910 to 1950, there was an increase in the use of E. coli strains like K12 and B due to progress in molecular biology. For example, in 1922, at Stanford University, K12 strain was isolated from a stool sample of a patient who was recovering from diphtheria. Charles E. Clifton and Edward Tatum used this strain to study nitrogen metabolism and tryptophan biosynthesis. Following that, Joshua Lederberg, a student of Edward Tatum, did an experiment and found that conjugation of E. coli allows them to exchange genetic information. The name of this famous experiment was the Lederberg and Tatum experiment. Another example is strain B. The proper history of this strain is not very clear. As far as we know, in the early days of phage research around 1918, a scientist named Félix Duhamel in France stumbled upon a particular E. coli strain while working at the Pasteur Institute. This bacterial workshop, then called Bacillus coli, became a hot potato among phage researchers. It was passed from scientist to scientist across France and eventually to the USA. Jules Baudet, a Belgian microbiologist, shared it with his student, and it eventually reached Martha Olstein at Rockefeller Institute in the US. She in turn passed it to Jacques Bronfenbrenner. By the 1940s, this well-traveled E. coli strain landed in the hands of Max Delbrück and Salvador Luria. The scientists then used it and discovered the famous Luria-Delbrück experiment, aka Fluctuation Test. Later, in 1958, Matthew Meselson and Franklin Stahl also used this strain to show that DNA replication is semi-conservative. We're not going to the details of these theories and experiments because that will make the video lengthier. Now come to the early days of 1960s. At that time, scientists gathered a good amount of knowledge on molecular biology using E. coli. The structure of DNA and its many properties were available at that time. Around 1965, Werner Arber, a Swiss microbiologist and geneticist, discovered types of particular enzymes that can digest phage DNA. These enzymes are called restriction enzymes since they can restrict the virus from entering the bacterial genome if that virus has a particular segment of DNA sequence or restriction site. This discovery consequently initiated the idea of genetic engineering, followed by recombinant DNA technology. The first people to work on this were at Stanford University and UCSF in the early 1970s. In 1980, Herbert W. Boyer and Stanley N. Cohen got a patent for human insulin that was made with E. coli. This made everyone understand how useful these colon dwellers can be. Within the 1980s, biotech medicines like Humulin, Intron A, Roferon, Humatrop got approval. Today, around 30% of biopharmaceuticals are getting produced by E. coli. In conclusion, we can see within 100 years, this bacterium came from infant guts, contributed significantly in microbiology, biochemistry, molecular biology, and finally ended up in genetic engineering and biotechnology that solved problems like diabetes, cancer or leukemia, hepatitis and growth hormone deficiency, and many more. But this is just a glorified good history. Now let's get back to the past and explore the bad parts. Okay, let's start again. It was around 1885, Theodor Escherichia isolated a bacteria from infant guts. He named it Bacterium coli commune. However, within a few years, a book named The Principles of Bacteriology reported that this bacterium has the potential of causing disease when present at other body sites rather than colon. But after several decades, scientists started to observe something different in the colon also. Most probably, a scientist named Adam first reported a relation between Escherichia coli and infant diarrhea in 1927. I couldn't find the paper, but this is the reference. The earliest paper I got from the internet is from John Bray, who used to work in the pathology laboratory of the Emergency Medical Service of London. From March 1943 to April 1944, they recorded 51 infants with summer diarrhea with 20 deaths. From postmortem, they observed that the intestines of these dead patients were dilated with very little inflammation. By that time, McConkey agar was already available. This type of agar can distinguish E. coli because E. coli can ferment lactose, and this feature provides the bacterial colonies pink or red color. Non-lactose fermenters cannot produce this color on McConkey. So John Bray and his team observed something weird that time. Usually, diarrhea-causing pathogens are non-lactose fermenters, such as Salmonella, Proteus, and Shigella. However, these pathogens are lactose fermenters. A study found a bacteria called the Bacteria coli napolitanum that takes longer to break down a certain sugar called maltose. This bacteria seems to be very common in the babies with severe diarrhea during the summer, but rarely found in healthy babies. They also identified a particular O-type antigen in this bacteria, but they couldn't identify the link between this pathogen and the diarrhea at that time. However, a contrasting report emerged from Africa in 1952. Researchers conducted a study on the causes of summer diarrhea in Bantu infants in Johannesburg. They analyzed stool samples from 20 infants who were hospitalized due to diarrhea and vomiting. They were conducting tests to detect specific strains of E. coli bacteria that had previously been associated with outbreaks of diarrhea in infants in other countries. According to the findings, the researchers determined that the specific E. coli strains were highly unlikely to be a significant cause of summer diarrhea in Bantu infants in that area. This indicates that other factors may be responsible for the diarrhea outbreaks observed in other regions. However, by the time a paper was published from the USA, and it might sound horrible to some of you what I am going to say next. So what researchers did in that study was they made healthy adults drink milk containing either a bacterial strain linked to diarrhea in infants or a normal harmless bacterial strain. When they gave the adults very large doses of 9 billion or 6.5 billion of the diarrhea-causing bacteria, many of them got food poisoning symptoms like diarrhea, vomiting, stomach cramps, and fever. But when given smaller doses of 530 million or 7 millions, the adults only had mild issues or no problems at all. Interestingly, most adults who drank the diarrhea bacteria, even the lower doses, developed antibodies in their blood. Even some who didn't feel sick, suggesting their bodies still recognized and responded to the bacteria. In contrast, the adults who drank 9 billion of normal harmless bacteria stayed completely healthy with no antibody response. Some adults carried the diarrhea bacteria in their intestines temporarily after ingesting it, but it didn't spread to their bloodstream or urine. The study showed that while healthy adults can usually fight off small amounts of these diarrhea germs, very high doses of billions of bacteria can make them ill, and they could potentially spread the bacteria to vulnerable babies. Okay. So this is the early 1950s, and now we know that some E. coli strains are bad, but not all of them are harmful. So the next question is, which strains are bad? Let's start again. In 1955, a study from Ohio reported that E. coli O127B8 is associated with acute diarrhea in infants. After a long time, in 1971, a study showed something interesting. Some scientists took 2 strains of E. coli bacteria that were isolated from American soldiers in Vietnam who had diarrhea and intestinal inflammation. They tested these bacterial strains with guinea pig, monkey, rabbit, and human. The researchers found that one group of E. coli strains produced a toxin that, when given to the volunteers, caused severe watery diarrhea resembling cholera. The other group of E. coli strains didn't make a toxin but were able to invade and penetrate the intestinal lining cells. When these strains were given to volunteers, they caused a dysentery-like illness with bloody diarrhea, painful bowel movements, high fevers, and toxic effects on the body. So in summary, these Vietnam bacterial strains highlighted 2 distinct ways E. coli can cause diarrheal disease, either through a cholera-like toxin mechanism or by invading the gut lining. Now, by 1970s, scientists started to realize that some strains of E. coli are pathogenic and the patterns of pathogenesis might vary. Some strains produce toxin and some invade intestinal cells. Another team showed several E. coli strains produce cytotoxicity in 1977. However, in 1978, a paper was published in Lancet that showed some E. coli can cause diarrhea without producing toxins or being non-invasive to the intestinal cell. Around that time, some researchers started to analyze the genome of pathogenic E. coli with the help of our old friend E. coli K12. So by this time, many researchers were going on. Some scientists were exploring how E. coli behaves in chicken. Some scientists also purified the E. coli toxin. But in 1982,
Something devastating happened.
47 patients from Oregon and Michigan were admitted to the hospitals. Their symptoms were severe crampy abdominal pain, initially watery diarrhea followed by bloody diarrhea, and little or no fever. All of them had a history of eating fast food from the same restaurant chain. The major feature of this outbreak was hemorrhagic colitis. Moreover, this E. coli was neither toxicogenic nor invasive. Serotyping revealed that it was E. coli
O157H7.
Sounds familiar? It is a very popular pathogenic strain. The possible source of this outbreak was most probably undercooked meat.
People infected with the E. coli bacteria, more than 150 people have become ill.
10 years later, from December 1992 to February 1993, another outbreak of O157H7 took place with 732 confirmed cases, 178 severe cases, and 4 deaths. It's called Jack-in-the-Box E. coli outbreak. It reached a total of 73 Jack-in-the-Box restaurants across California, Idaho, Washington, and Nevada. It became widely known as the most notorious food poisoning incident in recent times. The majority of individuals who suffered were under the age of 10. The outbreak originated from contaminated beef products served at Jack-in-the-Box restaurants. But it was not the end. In 1996, Odwalla E. coli O157 outbreak killed a 16-month-old girl and sickened 70 people. Source was apple juice. In 2000, Walkerton, Ontario faced another O157 outbreak with 2,000 plus cases and 7 deaths. Source was contaminated drinking water supplies. In 2005, South Wales E. coli O157 outbreak had 157 cases and 1 death. Source, contaminated vacuum packing machine. And it goes on. Just see this graph. But the largest outbreak by far we have seen took place in 2011.
Fears are growing that an E. coli breakout in northern Germany, which has now claimed 14 lives.
2 more deaths have been reported this morning after an E. coli outbreak
in
Germany.
The total confirmed cases were 3,950 and 53 patients died. The source of this outbreak was fenugreek sprouts. But most shockingly, this time, O157 was not the main villain. This time, scientists saw a new strain called O104H4. The outbreak, initially believed to be caused by an enterohemorrhagic or EHEC strain of E. coli, like O157, but further investigations revealed that it was actually caused by an enteroaggregative E. coli or EAEC. In 2023, we also faced another outbreak of E. coli in Calgary with 39 cases. And this outbreak was most probably a non-O157 outbreak. It was a Shiga toxin producing E. coli. Sometimes it's called ESTEC. So what is this EHEC, ESTEC, and others? Scientists have classified E. coli according to their pathogenesis. For example, enterotoxicogenic E. coli or ETEC produce toxin. Some of these toxins are similar to cholera-like toxin. It is the most common cause of traveler's diarrhea. 2. Enteropathogenic E. coli, EPEC, causes watery diarrhea. Their virulence is similar to Shigella. They don't have toxins like ETEC. Enteroaggregative E. coli, EAEC, can produce hemolysin. 4. Enteroinvasive E. coli, EIEC, can cause bloody diarrhea. It is similar to shigellosis. 5. Enterohemorrhagic E. coli, EHEC. O157H7 is EHEC. They might cause hemolytic uremic syndrome and sudden kidney failure. It produces Shiga toxin that can elicit an intense inflammatory response. 6. Adherent invasive E. coli, AIEC. This pathotype can replicate intracellularly. They are associated with Crohn's disease. Okay, so we have seen ESTECs. ESTECs are Shiga toxin producing strains. EHEC is a member of ESTEC group. Usually, pathogenic E. coli strains contains any of these genes. Moreover, there is a uropathogenic E. coli, UPEC, that causes urinary tract infection, or neonatal meningitis, NMEC type E. coli, that causes meningitis. Some are related to animal diseases like mastitis or sepsis. So you might think, okay, E. coli is harmless most of the time, and if you get infected by the pathogenic strain, by any chance you will go to the doctor and he or she will prescribe you antibiotics, then you will get well, right? Okay, here comes the ugly truth.
Superbugs.
Superbugs. They're superbugs.
They hang overuse, misuse, and the climate crisis.
However, antibiotics have been losing their punch against some types of bacteria.
So this part will be very short. If you carefully observe these types of studies, you will realize that both pathogenic and non-pathogenic strains of E. coli are getting resistant to different antibiotics, especially in lower and middle income countries where proper regulation of antibiotics is very challenging. All E. coli strains are getting exposed to antibiotics from foods, environments, or medicines. Like other bacteria, this is making non-harmful E. coli resistant to antibiotics along with pathogenic strains. Interestingly, scientists are using E. coli to study antibiotic resistance and pathogenesis. So, in that sense, we can say that we are using E. coli to fight E. coli. Lastly, I can say that none of the E. coli strains are good or bad. According to evolution and nature, they want to survive and reproduce. For pathogenic E. coli, our immune system cannot handle them properly, like non-pathogens. Hence, they initiate immune response and create diseases. We should be more careful about eating and handling raw fruits, vegetables, meat, and water. And we should take antibiotics in an appropriate manner with the help of doctors if E. coli infect us. Thank you.